Bioprinting is a rapidly evolving field that combines biology and engineering to create functional tissues and organs. This revolutionary technology holds immense potential for regenerative medicine, personalized therapy, and drug development. It is pushing the boundaries of healthcare and transforming the way we approach medical treatments. The image showcases a close-up of a 3D printed object made of biocompatible materials with a sleek, futuristic design and soft, ambient lighting that highlights the intricate details and delicate textures. This visual representation helps illustrate the advanced capabilities and precision of bioprinting technologies. Bioprinting is an advanced 3D printing technology that uses biocompatible materials, living cells, and bioinks to create functional tissues and organs. Biomaterials are engineered materials that support cell growth and function. These are critical components in bioprinting. Bioinks are cell-laden materials used directly in the printing process. The composition of bioinks is key to creating viable printed tissues. Printing systems are sophisticated machines that deposit bioinks in a controlled manner to build up 3D structures layer by layer. The timeline on this slide provides a brief history of the development of bioprinting technology. It covers the key milestones, starting with the early concepts in the 1990s, followed by the first successful bioprinted tissues in 2003. The 2010s saw significant advancements in bioprinting, including new biomaterials, bioinks, and printing systems. Now, the field is moving towards commercialization, with companies developing bioprinted products for applications like regenerative medicine and drug discovery. This visual timeline gives the audience a clear understanding of how bioprinting has evolved over time and where the technology is heading. Bioprinting is an exciting field that combines biology, materials science, and 3D printing to create living tissues and organs. One key aspect is biocompatibility. The materials used must be compatible with living cells, not causing any immune response or toxicity. Another critical factor is maintaining cell viability throughout the printing process. Keeping the cells alive and healthy is essential for creating functional tissues. Bioprinting also aims to mimic the natural structure and function of tissues and organs, creating a biologically relevant environment for the cells. This combination of biocompatibility, cell viability, and biomimicry is what makes bioprinting such a promising approach for regenerative medicine and tissue engineering. Bioprinting is an advanced technology that allows us to 3D print living tissues and organs. The key components of a bioprinting system include biomaterials, bioinks, cells, and the printing systems themselves. Biomaterials like hydrogels, polymers, and ceramics provide structural support and promote cell growth. Bioinks are cell-laden materials that are printed layer by layer to build up tissues and organs. The living cells are the building blocks that differentiate into various cell types, forming the basis of the tissues. The sophisticated printing systems precisely deposit the bioinks in a controlled manner, allowing for the creation of complex structures. All of these components work together to make bioprinting a powerful tool for regenerative medicine and tissue engineering. Biomaterials are a crucial component in bioprinting, as they provide the scaffolding and support for cells to grow and form functional tissues. Natural biomaterials like collagen, alginate, and chitosan are derived from living organisms and offer excellent biocompatibility, providing a natural scaffold for cells to thrive and promote tissue integration and regeneration. Synthetic biomaterials, such as polymers, hydrogels, and ceramics, are engineered to have tunable properties that can be tailored for specific applications. They offer flexibility in controlling mechanical properties, degradation rates, and cell interactions. Composite biomaterials combine the advantages of both natural and synthetic materials, leveraging their unique properties to create biocompatible and functional scaffolds. These materials can be tailored to provide the desired mechanical strength, biodegradability, and cell attachment for specific tissue engineering needs. The choice of biomaterial is crucial in bioprinting, as it directly impacts the success and functionality of the printed tissue or organ. Bioinks are the key materials used in 3D bioprinting to create living tissues and organs. The choice of bioink materials is critical. They must be biocompatible and support cell growth and function. Factors like printability, cell viability, and degradation rates are influenced by the bioink formulation. Viscosity and rheological properties of the bioink are also important for smooth extrusion and deposition during printing. The bioink needs to protect cells during the printing process and provide a suitable environment for cell growth and proliferation. Minimizing shear stress on cells and maintaining high cell viability are key considerations in bioink design and optimization. Fibroblasts are essential for producing the extracellular matrix, which provides structural support and function to tissues. Epithelial cells form protective barriers and linings in organs and tissues, playing a crucial role in organ function. Endothelial cells line blood vessels, facilitating the transport of nutrients, oxygen, and other important molecules throughout the body. Muscle cells generate contractile force, enabling movement and proper function of organs. Neural cells transmit electrical signals, enabling communication and coordination within the nervous system. Stem cells have the unique ability to differentiate into various cell types, offering exciting therapeutic potential in regenerative medicine. Tissue engineering is the process of creating functional tissues to repair or replace damaged areas of the body. This involves replicating the structure and function of the target tissue, like skin, cartilage, or bone. Bioprinting is a key technology in tissue engineering, allowing us to precisely place cells and other materials to construct these tissues. The ultimate goal of organ fabrication is to print whole organs, like kidneys, livers, and hearts, for transplantation. This is an incredibly complex process, requiring intricate designs and very precise placement of different cell types. Significant progress has been made, but there are still many challenges to overcome before we can reliably print functional organs for transplantation. Extrusion-based bioprinting is a key technique in the field of tissue engineering and regenerative medicine. The process involves a syringe-like nozzle that precisely extrudes a bioink, which contains living cells and biomaterials. This bioink is deposited layer by layer, building up a 3D structure. As the layers are deposited, they solidify, forming a scaffold that supports the growth and differentiation of the cells. This approach allows for the creation of complex, customized tissue structures that can be used for a variety of medical applications. The precision and control of the extrusion process is critical to ensuring the final construct has the desired properties and functionality. Inkjet bioprinting is a technique that uses a nozzle to spray tiny droplets of bioink containing cells and biomaterials onto a substrate. This method allows for precise placement of the droplets, creating a layer-by-layer buildup of the desired 3D structure. Inkjet bioprinting offers high resolution and precise cell placement, making it suitable for printing delicate structures and complex geometries. Some key advantages of this technique include high resolution and precision, controlled cell placement, and versatility in the range of biomaterials and cell types that can be used. However, there are also some limitations, such as potential cell damage, material compatibility issues, and challenges with scalability for larger tissues or organs. Overall, inkjet bioprinting is a powerful tool for creating intricate and complex 3D tissue structures with precise control over cell placement and biomaterial composition. Laser-assisted bioprinting is a highly precise technique that allows for the creation of intricate patterns and delicate structures. The non-contact nature of the laser helps minimize cell damage, which is crucial for maintaining cell viability and tissue functionality. However, this technique requires specialized equipment and expertise, making it relatively expensive and complex to implement. Additionally, the laser can potentially alter the properties of the biomaterials used, so careful optimization is necessary for specific applications. Laser-assisted bioprinting is a highly precise technique that allows for the creation of intricate patterns and delicate structures. The non-contact nature of the laser helps minimize cell damage, which is crucial for maintaining cell viability and tissue functionality. However, this technique requires specialized equipment and expertise, making it relatively expensive and complex to implement. Additionally, the laser can potentially alter the properties of the biomaterials used, so careful optimization is necessary for specific applications. Microfluidics-based bioprinting offers precise control over fluid flow, enabling us to carefully deliver cells and bioinks to create intricate 3D structures with high resolution. This technique minimizes cell damage during the printing process, which is a key advantage over other bioprinting methods. However, the specialized manufacturing required for microfluidic devices can make them expensive to produce. Scaling up microfluidic bioprinting to create large-scale tissues or organs can also be challenging, something we'll need to consider as the technology develops further. Bioprinting techniques have evolved to include hybrid approaches that combine the strengths of different methods. This allows for enhanced resolution, cell viability, and material versatility compared to single-technique approaches. For example, inkjet printing can achieve high resolution, while extrusion printing is better suited for larger structures. However, integrating these diverse technologies requires complex design and optimization, which can increase costs and technical complexity. Ensuring compatibility between materials and printing processes is crucial for creating functional and biocompatible structures. Cell damage is a key challenge in bioprinting. The printing processes can subject cells to mechanical stress, impacting their viability. Lack of vascularization in bioprinted constructs is a problem, as it limits the delivery of nutrients and oxygen to the cells. The biomaterial scaffold used in bioprinting may not provide the optimal conditions for cell attachment and growth. Another challenge is ensuring the bioprinted cells differentiate and function properly within the engineered tissue. Vascularization is critical for bioprinted tissues to survive and thrive. Without a functional vascular network, cells in the interior of the tissue will not receive the oxygen and nutrients they need. This can lead to cell death and prevent the tissue from fully integrating and functioning properly. Replicating the complex, intricate structure of blood vessels is a major technical challenge for current bioprinting methods. We need to find ways to incorporate vascular networks directly into the bioprinting process to overcome this hurdle. Developing solutions for robust, perfusible vascular networks is a key focus area for advancing bioprinted tissue engineering. Vascularization is critical for bioprinted tissues to survive and thrive. Without a functional vascular network, cells in the interior of the tissue will not receive the oxygen and nutrients they need. This can lead to cell death and prevent the tissue from fully integrating and functioning properly. Replicating the complex, intricate structure of blood vessels is a major technical challenge for current bioprinting methods. We need to find ways to incorporate vascular networks directly into the bioprinting process to overcome this hurdle. Developing solutions for robust, perfusible vascular networks is a key focus area for advancing bioprinted tissue engineering. The scale and complexity of bioprinting systems is impressive, showcasing the precision and detail required for this technology. Scaling up bioprinting to meet clinical demands is a key challenge, requiring high-throughput printing systems that can produce large quantities of bioprinted tissues and organs. Consistent supply of high-quality, biocompatible materials is critical for large-scale bioprinting, with stringent quality control measures in place. Obtaining enough cells for large-scale bioprinting is a significant challenge, requiring efficient cell culture and expansion methods to generate the necessary cell sources. Reducing the overall costs associated with bioprinting is crucial to making bioprinted products accessible and affordable for patients. Bioprinting is a rapidly advancing field with immense potential, but it also faces significant regulatory challenges that must be addressed. Ensuring the safety and efficacy of bioprinted tissues and organs for human use is of paramount importance. Rigorous testing and approval processes are necessary to protect patient health and well-being. Establishing robust quality control standards for bioprinting processes and materials is essential to maintain consistency and reliability in the final products. Ethical considerations around bioprinting, such as informed consent and potential misuse, must be carefully addressed to ensure the technology is used responsibly and ethically. Developing a clear regulatory framework for bioprinted products is vital for clinical adoption and commercialization, providing a clear path for developers and giving patients confidence in the safety and legality of these innovative treatments. The key goal of bioprinting is to create tissues that closely mimic the mechanical properties of natural tissues. This includes matching the elasticity, stiffness, and strength characteristics of the target organ or tissue. Achieving these desired mechanical properties is a significant challenge in bioprinting. The bioink composition, printing techniques, and post-processing methods all impact the final mechanical properties of the bioprinted tissue. Careful optimization of these factors is required to produce bioprinted tissues with the right mechanical behavior. Bioprinted tissues often lack the complex organization and functionality of natural tissues. The lack of vascularization limits nutrient and oxygen delivery, affecting cell growth and differentiation. Achieving appropriate cell-cell interactions and proper extracellular matrix formation is crucial for tissue maturation. The bioprinted scaffold must provide a suitable environment for cells to grow, differentiate, and integrate into functional tissues. Researchers are exploring strategies to enhance tissue maturation, including incorporating growth factors and using biocompatible materials that promote cell differentiation. Developing bioprinting techniques that create more complex vascular networks is a promising approach. The use of stem cells and the integration of bioreactors to mimic the natural environment are also promising for achieving tissue maturation in bioprinting. Bioprinting is an exciting technology that can create living tissue for medical applications. One key application is skin grafts for burn victims and patients with skin conditions. Bioprinted skin grafts can promote faster healing and reduce scarring compared to traditional skin grafts. Another application is cartilage repair. Bioprinted cartilage grafts can restore joint function and reduce pain for patients with osteoarthritis or other cartilage injuries. Bioprinting can also create bone scaffolds that promote bone growth and healing in patients with fractures or bone defects. These bioprinted tissues and grafts have the potential to significantly improve outcomes for patients with a variety of medical conditions. Bioprinted liver models provide a platform for testing drug toxicity and efficacy, mimicking human liver function. Bioprinted heart models allow researchers to study drug effects on cardiac cells, evaluating potential cardiotoxicity and assessing drug safety. Bioprinted skin models offer a human-relevant platform for evaluating the potential skin irritancy of new drugs and chemicals. These bioprinted tissue models enable more accurate and reliable preclinical drug testing compared to traditional cell cultures or animal models. By using human-derived cells, these bioprinted tissues closely mimic the real physiological responses, leading to better predictions of drug safety and efficacy. Bioprinting is a powerful tool that is transforming drug development and toxicology testing, helping to bring safer and more effective medicines to patients. Bioprinting is a powerful technology that allows us to create personalized medical solutions. One key application is patient-specific models. We can use bioprinting to create tissue models that perfectly match a patient's genetic makeup. This enables us to test treatments and drugs on these personalized models, leading to more tailored and effective care. Another important application is organ replacement. Bioprinting can be used to create organs that are a perfect match for the patient, reducing the risk of rejection and improving outcomes. Bioprinted tissues can also be used to test the effectiveness of different drugs for a particular patient. This targeted therapy approach allows us to identify the most effective treatments, leading to better outcomes. Overall, bioprinting is revolutionizing personalized medicine by enabling us to create customized solutions that are precisely tailored to each individual patient. Bioprinting is an exciting technology that allows us to create highly detailed, realistic 3D models of human organs. These models provide invaluable tools for research, education, and drug development. They offer a much deeper understanding of human anatomy, physiology, and disease processes compared to traditional 2D diagrams or physical models. The models showcased here demonstrate the incredible level of detail that can be achieved, from the intricate blood vessels in the heart, to the complex neural networks in the brain, to the delicate alveoli structures in the lungs. These organ models are created using advanced bioprinting techniques, which leverage 3D printing to precisely build up layers of biological materials. The futuristic laboratory setting emphasizes how this technology is pushing the boundaries of what's possible in the field of medicine and healthcare. Bioprinted skin grafts are a revolutionary technology that can help heal wounds and promote tissue regeneration. These grafts contain living cells and biomaterials that provide a protective barrier and facilitate the growth of new, healthy skin. Bioprinted grafts can accelerate wound closure, reducing healing time and minimizing the risk of infection. The precision of the bioprinting process also allows for improved aesthetic outcomes, helping to minimize scarring, especially for severe wounds like burns. The potential of this technology to transform wound care and tissue regeneration is truly exciting. Bioprinting offers exciting applications for repairing cartilage and bone injuries. For cartilage repair, we can create biocompatible grafts using materials like collagen and hyaluronic acid. These grafts can help restore joint function and reduce pain for patients with osteoarthritis or other cartilage damage. Bioprinting also allows us to create personalized bone scaffolds to promote healing in patients with fractures or bone defects. These scaffolds can be tailored to the patient's specific needs, providing a more customized treatment solution. Overall, bioprinting is a powerful tool for regenerative medicine, helping patients recover from debilitating cartilage and bone injuries. Bioprinting offers exciting possibilities for addressing cardiovascular issues. We can use bioprinting to create functional blood vessels for vascular grafts and bypass surgeries. This could provide a solution for patients with peripheral artery disease or coronary artery disease. Bioprinting can also be used to create custom heart valves for patients with valve defects. This offers a less invasive alternative to traditional valve replacement surgery. Additionally, bioprinted cardiac patches can be used to repair damaged heart tissue. These patches can promote healing and reduce the risk of heart failure. Neural tissue engineering using bioprinting offers valuable opportunities for research and drug discovery. We can create 3D models that mimic the complex structure and function of the nervous system. These models can provide important insights into neurological diseases and disorders. However, there are significant technical challenges to overcome, such as maintaining the viability of cells during the bioprinting process, developing functional vascular networks to support the tissue, replicating the intricate circuitry and connectivity of the brain. Bioprinted liver models are a powerful tool for drug testing, disease modeling, and personalized medicine. These models allow us to better understand liver function and disease processes, which can lead to the development of new drugs and therapies. However, there are some key challenges in bioprinting liver tissue, such as replicating the complex liver structure and maintaining cell function over time. Scaling up bioprinting to meet clinical demands will require further advancements in areas like bioink formulation, printing techniques, and cell culture methods. Bioprinted kidney models offer a valuable platform for drug testing and disease modeling, helping researchers understand kidney function and disease processes. These models also hold potential for personalized medicine, allowing researchers to tailor treatments to individual patients. However, there are significant challenges in bioprinting kidney tissue, including replicating the complex kidney structure, maintaining cell function, and developing suitable biocompatible materials. Vascularization remains a major challenge, requiring further research to create functional blood vessel networks within the bioprinted tissue. Bioprinted kidney models can be used to study drug toxicity, test new therapies, and develop personalized treatments for kidney diseases. In the future, bioprinting might enable the creation of bioengineered kidneys for transplantation, offering a solution for patients with end-stage renal disease. Bioprinted pancreas models provide a valuable platform for studying insulin production, diabetes research, and drug development. These models can be used to test new therapies and improve our understanding of the underlying mechanisms of diabetes. This could potentially lead to the discovery of new treatments for this disease. However, there are significant challenges in bioprinting pancreatic tissue, such as replicating the complex structure of the organ, maintaining cell function, and developing suitable biomaterials. One of the biggest hurdles is creating functional blood vessel networks within the bioprinted tissue, which is crucial for the tissue's survival and integration. Corneal blindness is a major global health issue, affecting millions of people worldwide. Bioprinted corneal tissues offer a promising solution to replace damaged or diseased corneas and restore vision. Key advantages include the ability to create customized tissue grafts that can integrate with the patient's own tissue. However, there are still some technical challenges that need to be addressed, such as ensuring adequate vascularization of the bioprinted tissue, maintaining cell viability during the printing process, achieving proper integration between the bioprinted tissue and the surrounding host tissue. Further research is needed to optimize the bioink formulation, printing techniques, and post-printing procedures to overcome these challenges. With continued advancements in this field, bioprinted corneal tissues have the potential to significantly improve treatment options and outcomes for patients suffering from corneal blindness. Bioprinting is an exciting new medical technology, but it raises important ethical considerations that we must carefully address. Informed consent is critical. Patients must fully understand the risks and benefits before agreeing to a bioprinting procedure. Clear and comprehensive information is essential. Privacy and data security are also crucial. The collection and use of patient data must be carefully regulated to protect individual privacy and ensure responsible data management. Access and equity are important. We must ensure these innovative therapies are accessible to all patients, regardless of socioeconomic status. Bioprinting also raises complex social and ethical questions, such as the potential for genetic engineering, the implications for human identity, and the role of society in shaping the future of this technology. These are important issues we need to thoughtfully consider. Bioprinting innovations can be protected through patents covering novel printing techniques, biomaterials, and bioinks. Companies can register trademarks for their bioprinting products and services, distinguishing them in the market. Bioprinting software, designs, and manuals can be protected by copyrights, safeguarding creative works. Confidential information about bioprinting processes, formulations, and data can be protected as trade secrets. These various forms of intellectual property are crucial for commercializing bioprinting technologies and maintaining a competitive edge. Government agencies like the NIH and NSF provide funding to support promising bioprinting research projects that advance scientific knowledge and address critical healthcare needs. Venture capital firms invest in startups and companies developing innovative bioprinting technologies and applications, fueling the growth of the industry. Pharmaceutical companies, medical device manufacturers, and other industry players collaborate with research institutions, providing funding and expertise to accelerate the development and commercialization of bioprinting. Foundations and individual donors contribute to bioprinting research, supporting projects that address specific diseases, advance ethical considerations, or promote public awareness of this transformative technology. Bioprinted products must go through rigorous preclinical testing in animal models to evaluate safety and efficacy before moving to human clinical trials. Clinical trials are required to assess the safety and effectiveness of bioprinted products in treating specific medical conditions in human patients. Regulatory approval from agencies like the FDA is necessary before bioprinted products can be commercialized and made available to patients. Continuous post-market surveillance is conducted to monitor the long-term safety and efficacy of bioprinted products in real-world settings. Bioprinting research requires diverse expertise from fields like biology, engineering, materials science, and medicine. Multidisciplinary teams bring different perspectives to address the complex challenges of creating functional tissues and organs. Collaborating with industry partners like pharmaceutical companies and medical device manufacturers is crucial for translating bioprinting research into clinical applications. These partnerships provide resources, expertise, and market access to support the development and commercialization of bioprinting technologies. Bioprinting research is increasingly global, with international collaborations facilitating knowledge sharing, resource pooling, and advancing the field worldwide. Organovo is a leading bioprinting company that specializes in 3D printed human tissues and organs, such as liver and kidney models, for research and drug development. BioLife 4D is a bioprinting startup focused on developing bioprinted organs for transplantation. Their unique bioink formulation mimics the natural environment of living cells to promote cell survival and function in the printed organ. Cellink is a global bioprinting company that develops and supplies bioinks, bioprinting equipment, and services for researchers and companies worldwide. They are known for their commitment to open-source platforms and collaboration in advancing the field of bioprinting. The Weiss Institute at Harvard University is a leading research center in bioprinting for tissue engineering and regenerative medicine. They focus on developing biocompatible materials, bioinks, and advanced printing techniques to create functional tissues and organs. The Wake Forest Institute for Regenerative Medicine has made significant breakthroughs, including the world's first bioprinted human organ. Their research aims to advance tissue engineering, organ transplantation, and personalized medicine through bioprinting. The UCSD Center for Bioprinting is focused on innovative bioprinting technologies for functional tissues and organs. Their work involves bioink formulation, printing techniques, and new applications in regenerative medicine, drug discovery, and disease modeling. The Institute for Bioengineering of Catalonia, IBEC, in Barcelona is a leading bioprinting research center with a focus on musculoskeletal, cardiovascular, and neural tissues. IBEC develops new biomaterials, bioinks, and printing methods for creating functional tissues and organs. The World Bioprinting Summit is an annual event that brings together industry leaders and researchers to showcase the latest advancements in bioprinting. The Society for Biomaterials annual meeting is a comprehensive forum that covers biomaterials, tissue engineering, and bioprinting, with dedicated sessions on cutting-edge research. The International Conference on 3D Printing and Additive Manufacturing is a broader conference that includes a dedicated track on bioprinting advancements. The Bioprinting Workshop Series at the University of California, San Diego offers hands-on training on bioprinting techniques, equipment, and bioink development. These events provide valuable opportunities for the bioprinting community to network, share knowledge, and stay up to date on the rapidly evolving field. Bioprinting is an exciting and rapidly evolving field that is transforming the way we approach healthcare and tissue engineering. At the undergraduate level, many universities now offer courses and programs that provide students with a strong foundation in the key areas of bioprinting, including biomaterials, bioinks, and printing techniques. At the graduate level, specialized master's and PhD programs delve deeper into advanced bioprinting methodologies, tissue engineering, and organ fabrication, equipping students with highly specialized skills. For industry professionals, short courses and workshops offer practical, hands-on training on specific bioprinting technologies, equipment, and real-world applications. These educational programs are crucial for developing the next generation of bioprinting experts and driving innovation in this transformative field. Bioprinting is a rapidly evolving field with exciting developments on the horizon. One key trend is the enhancement of biomaterials to better mimic natural tissues, improving biocompatibility, degradation rates, and mechanical properties. Advancements in bioinks are also crucial, enabling the creation of more complex and intricate tissues with improved printability, cell viability, and biofunctionality. The integration of artificial intelligence can optimize bioprinting parameters, design complex structures, and analyze tissue development, leading to personalized and automated bioprinting. Multi-material bioprinting allows for the simultaneous printing of various biomaterials, enabling the creation of complex tissues with varying structures and properties, mimicking the complexity of natural tissues. Strategies for vascularization are essential to ensure adequate oxygen and nutrient delivery for cell viability and function within bioprinted tissues. Bioprinting holds great potential for personalized medicine, allowing for the tailoring of tissues and organs to individual patients based on their unique characteristics. This technology can also be leveraged for drug discovery and development, using bioprinted tissues and organs as models for testing drug efficacy, toxicity, and interactions. Bioprinting can contribute to regenerative medicine by creating functional tissues and organs for transplantation, addressing the shortage of organ donors. Bioprinting has the potential to revolutionize healthcare by creating functional tissues and organs for transplantation, addressing the shortage of organ donors. Personalized therapies and treatments tailored to individual patients' needs and genetic makeup can improve healthcare outcomes, reduce costs, and enhance quality of life. However, bioprinting also raises ethical and social challenges around access, equity, informed consent, and potential misuse that require careful consideration and discussion. Establishing ethical guidelines and regulatory frameworks is crucial to ensure the responsible use of this transformative technology. Public engagement and open dialogue are essential for shaping the future of bioprinting in a way that benefits society as a whole. The idea of printing tissues and organs has been around since the 1990s, when the concept first emerged. Over the years, bioprinting technology has advanced significantly, evolving from basic prototypes to sophisticated, complex systems. Bioprinting holds immense potential to revolutionize the field of regenerative medicine, enabling the fabrication of tissues and even entire organs. Looking to the future, bioprinting could help solve the critical shortage of organ donors and enable the creation of personalized therapies tailored to individual patients.